Hi, my name is Molly Wagner, and I'm here to present a poster for the Dybowski Group. For ease of viewing, I have converted our poster into a PowerPoint presentation, but the full poster should be available on whatever website you are viewing this video. My poster is entitled Understanding Soap Formation in Oil Paintings Containing Lead and Zinc Whites. This project has been done in collaboration with numerous groups, including the Metropolitan Museum of Art, the National High Magnetic Field Laboratory, and Brookhaven National Lab. And a vast majority of the project has been funded by the NSF. So we are looking at a reaction that occurs between heavy metal containing pigments such as lead white and zinc white and the free fatty acids that form in the oil binding media of oil paintings. So why is this important? Oil paints are extremely popular. They've been extremely popular for hundreds of years. If you walk into an art museum, the vast majority of things that you're going to see throughout there are going to contain or utilize oil paints in some way or another. And unfortunately, um, oil paints in their simplicity are some sort of pigment and some sort of oil binder, which are the two things that you need to potentially form these metal carboxylates. So, hidden in every single oil painting or anything containing oil paints is the potential for this destructive reaction to occur. So, unfortunately, these soaps are very problematic in terms of a conservation aspect. They can form in paintings in numerous ways, showing up as aggregates that protrude through the surface, giving almost a pot marked look to the surface of the painting. They can form cracks or hazy films, or they can even form between layers of paint, causing separation and delamination. Overall, this is just a conservator's nightmare. Um, so we are trying to approach the problem from the chemistry side of things. We want to learn what these soaps are, how they form, how they aggregate, um, and anything that we can learn that could potentially assist conservators in preventing or reversing this damage if at all possible. So I've included a picture here. You can see a small lead soap protrusion that is forming beneath the layers of the colored paint. So how pervasive is this problem? I've included here pictures of various paintings, span roughly 500 years, uh, quite a few different countries, quite a few different artists. Um, these were all done in different places at different times with different paints from different sources, and yet unfortunately they all exhibit soap formation. So how are we approaching this? Well, we're using a number of different methods and experimental techniques to try to holistically approach this problem. First off, we are using crystal structure determination by powder or single crystal X-ray diffraction to try to learn about the structure and the packing of these soaps. Our group primarily focuses on NMR. My predecessors did a lot of work in lead 207 NMR. My project focuses a lot on zinc 67 NMR. We also use proton and carbon 13 NMR as well to learn about as much of the soaps as we can. We also use FTIR as well as various synchrotron techniques to, again, try to elucidate as much information as we possibly can. The little video that just played is a 3D composite of tomography imagery of a lead white sample. So what you can see is the structure of the lead white pigment in the dried paint. Um, and at this small of a scale, it almost appears like a kitchen sponge. So you can see that there are numerous pores and channels and areas for various things to move throughout the paint matrix. So what do we know so far? So this is our 5-step process. Uh, to simplify everything down. Um, so step one, the free fatty acids have to form. Paint is a polymer, or at least oil paint is a polymer. As it dries, the unsaturated fatty acids in the oil matrix polymerize together. Saturated fatty acids cannot polymerize and therefore are excluded from the paint matrix, but they don't just sit there, and that moves us on to step two. So step two is the movement of reactants, whether or not it is just the free fatty acids that are moving about, if it's the free fatty acids and the metal ions, we are still working on elucidating that. Regardless, one way or another, these things have to move. Once they move, they come together, they react. Soap's formed. This is a problem. Small amounts of soap, while they are obviously not ideal, are not necessarily an issue immediately. But that brings us to step four. Step four, these small bits and pieces of soaps aggregate together to become large enough to get us to step five, which is irreversible damage becomes available. Figure B is our very cartoony proposed mechanism of soap formation. So based off of our experimental data, we suggest that the free fatty acids, which in this case are little orange squiggles, attack the pigment particles, the red circle. They form a layer of soap around the outside of the pigment particle, which then means that to finish the reaction, that the free fatty acids must diffuse through the formed soap to get to the remaining pigment particles. So what do we know about the coordination environment? So this is work done by my predecessors, Anna Murphy and Yao Yao. They looked at a series of lead carboxylates that are known to potentially show up in soap formation to try to discern the packing information and the structure of these lead soaps. And what you can see is that there is a definite grouping. The short chain carboxylates and the long chain carboxylates have different NMR spectra, indicating that there are two different packing groups for the lead carboxylates. Uh, Carbon-13 NMR as well as FTIR data also collaborate this data. We've also done kinetic studies by NMR, monitoring the reaction of a deuterated free fatty acid, in this case palmitic acid, on a model paint film. So you can see here I have included a NMR spectra of both palmitic acid and lead palmitate. And you can see that there is a difference in the peaks of the two. So you can watch over time as the carboxylate peak of the palmitic acid decreases and the peaks for the lead carboxylate increase, therefore allowing you to monitor the reaction over time. And so we have done this for the same quantity of acid and the same paint samples, but we have exposed the paint films to differing levels of relative humidity. You can see our data there in figure B, and figure C shows that there does seem to be a correlation between the amount of relative humidity and how fast this reaction actually occurs. So my project has been focusing a lot on Zinc NMR, which we are doing in collaboration with the Mag Lab down at the University of Florida. So much like the lead NMR, we have been looking at the differences in the series of zinc carboxylates that we have been looking at, and they show again that there is a divergence, but it does not occur in the same place as the lead NMR. Currently, we are working on a kinetic study similar to the lead kinetics that I showed on the previous slide, and that data should be published here in the near future. We've also done synchrotron studies to look at these various carboxylates. And we have shown that microzanes cannot differentiate the soaps from each other, but they can differentiate the soaps from the original pigments. Synchrotron techniques can also be used as imaging, as I showed you a couple of slides ago with the X-ray tomography data, which lets us determine the porosity and the pigment distribution in the paint films. We've also been utilizing a lot of single-sided NMR. Most of this work is done with our collaborators up at the Metropolitan Museum of Art in New York. Um, Single-sided NMR is extremely useful in conservation work. It is portable and does not require destruction of the sample in order to be able to use it, which is super awesome for conservation, as they generally frown upon you touching the actual artwork, um, which does make it a little difficult to study. So we have been looking at diffusion of water through model paint films, and what we can see is that there does seem to be two different diffusion coefficients, which we have theorized to be free water and constrained water. So again, going back to that image of the sponge-like appearance, the water can go through the different pores, moving somewhat freely in the open spaces between and being constrained in some of the smaller pores. As for future projects in this group, uh, obviously a continuation of the oil paint studies is a possibility. Our collaborators at the Met have also brought up the idea of looking at crazes and surface impurities on daguerreotypes. Uh, this could also be done potentially in collaboration with some of the surface chemists at the University of Delaware as well, using TOF sims. Um, Unfortunately, I do not know very much about this aspect of the project. That is a relatively new idea, but the Met does have a large collection of daguerreotypes, daguerreotypes being early essentially early photographs that utilized a iodine sensitized silvered plate and mercury vapor, and as these photos have aged, um, there have been surface essentially surface crusts that have begun to show over the surface. So learning what those crusts are and how best to remove and or prevent them would obviously be of great use to conservationists. Um, I hope that that poster slash presentation gave you a decent overview of what we have been doing in our group. If you have any questions, feel free to contact myself or Dr. Dybowski, and we would be happy to talk with you. Thank you so much for your attention.